Welcome. In the last session, we have seen how the neuron maintains polarized membranes. And so, to maintain the polarized membranes, we have understood that the membranes will have sodium voltage gated channels. So, the sodium voltage gated channels, and they also have potassium voltage gated channels, and along with them there are even sodium leakage channels. So there are sodium leakage channels and also potassium leakage channels, and lastly are sodium potassium pumps. So these are different proteins that are present on the membrane of a neuron, and especially we have seen in case of the axon. So the axon membrane, which we call it axolemma, shows the presence of these sodium voltage gated channels, potassium voltage gated channels, and sodium leakage channels, and even potassium leakage channels. And we also see the presence of sodium potassium pumps. Now, out of these leakage channels, what we see here, the potassium leakage channels are more in number. The potassium leakage channels are more in number than those of sodium leakage channels. So hence, with this, we understand that the axolemma the axolemma has greater permeability. It has greater permeability to potassium ions, greater permeability to potassium ions than to sodium ions. Right. So I told that the potassium leakage channels are more in number than those of sodium ions. And with all this, we have understood that it maintains the positive charge on outer side of the membrane and a negative charge inside of the membrane. So this is what we call a polarized membrane, right? Now in a resting membrane, means when it is not conducting any impulse, then we call it a resting membrane. So in a resting membrane, the potentials range from minus 40 millivolts to 90 millivolts, minus 90 millivolts. So it will range from minus 40 millivolts to minus 90 millivolts, and the typical value of a resting membrane potential. So the typical value of resting membrane potential that will be around minus 70 millivolts. So this is the one which we will consider the resting membrane potential. So the resting membrane potential is minus 50 millivolts. So the potential till it reaches to minus 55 millivolts, there is no chance of a depolarization, or the neuron transmits the nerve impulse. So here the neurons will follow all or none principle. So okay, all or none principle is followed by it will be followed by these neurons. So what it means that either it transmits the impulse or it doesn't transmit the impulse. So here to show this, actually the neuron should get a threshold value. It should get a threshold value to transmit the impulse. To transmit the impulse. If that threshold value is not reached, then the neuron will not show the transmission of the impulse. So this threshold value will be around minus 55 millivolts. So if it is under this, right, the neuron will not show, it will not transmit any impulse. So reaching this will make it to transmit the impulse. So that is what we say. So the neurons will follow all or none principle. Either they transmit or they don't transmit at all. So they will transmit the impulse only when the value reaches to minus 55 millivolts. So now let us see how that generation of the nerve impulse takes place and conduction of the nerve impulse takes place. So here I take the axon, right, and this is axolemma, and we know that the charge on the outer side is positive. So outer side is positive, and this is the ECF, the extracellular fluid, fluid, and inside this is the exoplasm. So exoplasm, and okay, so let's see the positive charge on the outer side of the membrane, and we have is the negative charge on inner side of the membrane. So the negative charge on inner side of the membrane. The fluid that is outside the axon, that is in ECF. So we know that in ECF, right, so there is low concentration of potassium ions. So it has a low concentration of potassium ions, and there is high concentration of sodium ions. Now inside the exoplasm, it's opposite to that. We know inside it has a high concentration of potassium ions and low concentration of sodium ions. Of course, along with this, there are negatively charged proteins. Here already we have seen it, so I'm not going to discuss about it because they are permanently located in the exoplasm, and we don't show any movement of those negatively charged proteins moving in and out of the exoplasm. So I'm not mentioning it. Okay, so here will mainly concentrate on sodium and potassium ions movement. So when there is a stimulus, the point where the stimulus is applied, it results in the opening of sodium ions. So it results in the opening of sodium ions. So you can see here. So these are sodium voltage gain voltage gated channels. Now this will result in movement of right. So here this is the stimulus which I have mentioned. So applying stimulus. So when stimulus is applied, it results in opening of this sodium voltage gated channels, and this will result in movement of the sodium ions into the exoplasm. So here sodium ions entering into the exoplasm, and this will result in charge reversal at that point. Okay, so say suppose this is a point A. So here this is a at this is a point A, and I will take here point B. So at point A, the opening of these voltage gated channels resulted in the movement of the sodium ions into the exoplasm, and this has resulted in the charge reversal at that point. So we have the positive charge inside and the negative charge outside. Now at this point there is a depolarization of the membrane, and this depolarization of the membrane is the one, okay, what we call the action potential. Okay, say suppose, so this is, I'm just mentioning at this point, so say this is the action potential. Action potential. Okay, so this is generated only when the value, okay, becomes minus 55 millivolts. So this is the threshold value. So till this threshold value reaches, okay, the membrane, I mean the axon will not transmit the nerve impulse, right? So now at right, so at this point we have negative charge outside and positive charge inside, and now the location ahead of this where I said that there is a site B, right? The positive charge is present outside and negative charge is present inside. So because of this, the current will flow from the site A to site B in the exoplasm. So in exoplasm, current flows from site A to site B. Okay, now in the ECF, okay, in ECF or on the outer surface of the membrane, on outer surface of membrane, the current flows from site B to site A. Current flows from site A to site, site B to site A. So this is where I'm showing from this point to this point. So this is the current flow. So this is the current flow. Now this is followed by the movement of potassium ions from inside, that is from the exoplasm to outside into ECF at the point of depolarization. Okay, so now immediately we'll see. Okay, so at this point we have is the potassium voltage gated channels. Okay, so this is potassium voltage gated channels. So I'll just mention with the okay, so in this way I'll just mention. So this is potassium voltage gated channel, right? And this will allow the movement of the potassium ions to outside. So it will allow the potassium ions to move outside at the point of depolarization. So this generates the action potential. Of course, that action potential is what we can also call it impulse, right? So the moment the sodium ions open, the sodium ions rust into the exoplasm, right? And it resulted in the depolarization of the membrane, and that will be immediately followed by the movement of potassium ions from the exoplasm into the ECF, and this will result in bringing back, okay, the charges at that point of depolarization back to normal. So now we can see in this case, okay, so now here the charge will come back, so plus here and minus. Now at the B side, okay, here again there is a movement of sodium ions into this. So again here there is movement of sodium ions into the exoplasm, and now here this side will become, okay, so at this point it becomes, okay, reverse. So you have the positive inside, okay, negative, same, the negative and positive. Now earlier the action potential which was present at site A, now it moves to site B. Now it moves to site B, and the point at A which was depolarized, now it will get repolarized. So now this point there will be no action potential present. The action potential moved to site B, and at this point it will become normal. So at the site A it comes back to normal state. That's what we call it polarized. At site A, it will become repolarized. Now, say suppose we have a site C. Now the same process, okay, takes place, and from here to from point B to, okay, to the site C, the current flows right inside, and on outside from C to B, from C to B, the current flows. And now, okay, the action potential. So here also there will be, okay, again sodium gated channels that will allow the movement of sodium ions into the exoplasm. Okay, so wherever I mention this in into the exoplasm, that indicates the movement of sodium ions, right? Of course, and at the same time here there will be, every time the sodium ions enter into it, it gets depolarized, and when the potassium ions move out, it will immediately become repolarized. So at B point also, the movement of the potassium ions back into this ECF, right, will make at that point to come back to the normal state. So now at that point again the charge comes back to, right, normal, right, so it comes back to, right, normal. So here also, right. So and here you don't find that action potential at the site A. Now even at the site B also the action potential has moved from the B point to C point. So at C point we have, okay, the charge positive inside, right, and the charge negative outside. So now at this point B there is no action potential. So now the impulse that started, okay, at site A, now it finally came at right, so it came to, okay, site C. So here is the action potential. So this will be continued, okay, that will get transmitted to next neuron. So that's an again a different part, how this information that is generated in this neuron will get transmitted to the next neuron. It may be through an electrical or okay, chemical synapses that we will see in a in coming sessions, right. So this is how the transmission of nerve impulses takes place, and I told that these nerves, they follow all or none principle. So they follow all or none principle. So where I told you that according to this all or none principle, the nerve impulse is either conducted totally. So it is either conducted totally. So that is nerve impulse is either conducted totally or it is not conducted at all, or it is not conducted at all. So that's what we call it all or none principle. So now here I'll show you an animation that how the transmission of nerve impulse takes place. So we know this is a neuron, right, and so this is how the impulse that taken from the dendrites passes to the cyton and then passes to the axon and then finally to the synaptic knobs, right. And so as I said, okay, so let's take this membrane here, and we know the outside of the membrane is positive and inside is negative, and you can see more sodium ions inside and less outside. So here is this sodium potassium pump, and I told that this is the one that is mainly responsible for maintaining the concentration differences right across the membrane. So we have seen that there will be more number of right, the sodium ions in the ECF and less in axoplasm, less in the axoplasm. Okay, so here we'll see. So that's the difference between interior and exterior of the cell, is called the resting membrane potential. So now what happens, so you can see these are sodium channels and this is a potassium channel. So you can see here, okay, the sodium ions moving into the axoplasm, moving into the axoplasm. Okay, so these are actually the gates which will open or close. So opening of one sodium ion, okay, will make the other sodium ions to open as they respond to the voltage stimuli. So you can see sodium ion entering in, and at this point it got depolarized. Okay, and then at the next point, so you can see and the action potential continues in a direction. And then immediately after this there will be opening of potassium channels. So here they're closed, and immediately potassium channel there gets open. Now the potassium ions will move outside, and that results in repolarized. So you can see, so at the point where the potassium ion moves, it comes back to the normal state. So this action potential, so how it moves all along the axon. Right. Now let us see this on a graph. We'll understand by taking a graph. So here, right, on the x-axis will take time. It's the time. And on the y-axis, I'm going to take the membrane potential. And that will take it in millivolts. So membrane potential in millivolts. Okay, and say suppose here, okay, at this point we have is minus 100 millivolts. Okay, and here is around, okay, minus 50 millivolts, and here, okay, we have 0, and then say at this point we have plus 50 millivolts. Right, now here, so I'm going to take a threshold value. Before that, I'm showing you here, this line indicates it is the resting potential. Okay, so this point is a resting potential, is resting potential. Okay, right. So now here, when the stimulus right takes up the neuron, so you can see this is of course in the beginning it is in the resting potential, and when it takes up a stimulus, it comes to a point of to a threshold value. So it comes to a threshold value. So here I'll just mention the threshold value. So this is, so in most of the mammalian neurons, the threshold value is minus 55 millivolts. So this is threshold potential, and it is minus 55 millivolts. So when, okay, so this is when it comes to, okay, that point, okay, so here after coming to this point, okay, so this is the threshold value, or I'll just mention with an, okay, and another color. So this is the threshold value. So this red color indicates the resting potential, okay, and this one indicates the threshold, and after this it results in, okay, increasing the value to, okay, so to this point, and then it comes down. So that point is the action potential, and then it comes down, right, and here we have an, okay, so from here I'll just mention then another color. So this is, okay, and then again it comes to the resting potential. Okay, and again it will continue, of course, this resting potential. So I'll put some okay numbers to them. We'll see here. Okay, of course this point here is the action potential. Okay, right, so this is action potential. So here I'll see, so I'll just mention here this is 1, and this I'll take it 2, and this is 3, and this is the 4. Okay, and this point is 5, and this point is 6. So now let us see what are all these 1 by 1. The first one, so that is 1, it is resting state. So this is the resting state. And coming to the second one is the depolarization state. So this is depolarization. Okay, and I'm sorry, it's a depolarization. And then third one is, right, it's a rising phase of the action potential. It's called the rising phase of action potential. Phase of action potential. And then the fourth one will be the falling phase. So that will be the okay falling phase of action potential. And then coming to the fifth point, so this one is the fifth one. This is the one what we call it undershoot. So later we'll see about this. And again coming to the sixth one, it will be a resting phase. So that will be the resting phase. So instead I'll say, instead of 6, we'll take this as 1 itself. So this will take okay as a 1 itself. So that comes back to the okay resting potential. So again, okay, a value of threshold okay value if it gets, and again there is action potential, raising of action potential, and this is how we see in a graph. Now coming to this action potential, and this action potential you can take it at plus 45 millivolts. So here I mentioned the threshold potential at minus 55 millivolts, and the action potential at plus 45 millivolts. And this resting potential will take it minus 70 millivolts. So here I mentioned something called undershoot. So this undershoot, okay, what actually it is. So, okay, so as the voltage falls below minus 70 millivolts, okay, that is the level of resting potential. So the voltage falls below, right, minus 70 millivolts, okay, that is to the level of the resting state. That is what we call this undershoot. So we have a small portion okay below the resting potential, and it is the undershoot, and then again it comes back to the normal the resting state, and the process continues.